The mitochondria contains an inner mitochondrial membrane and outer mitochondrial membrane, and cristae, which are foldings of the inner membrane. The first step in cellular respiration is glycolysis. Glycolysis takes place outside the mitochondria in the cytoplasm. In glycolysis, glucose, a 6-carbon molecule, is broken down into 2 molecules of pyruvate. Pyruvate are 3-carbon molecules. For 1 glucose molecule, 1 ATP is generated. Another thing that is made from glycolysis is NADH. Pyruvate diffuses into the mitochondria, and the pyruvate dehydrogenase complex converts the 3-carbon molecule pyruvate into acetyl-CoA, which is a 2-carbon molecule. Acetyl-CoA goes into the Krebs cycle, and during this process, carbon dioxide is given off. The Krebs cycle also gives off 2 ATP. Energy is added to NADH and FADH2. NADH and FADH2 have high energy electrons they can carry to the 3rd step of cellular respiration, which is the electron transport chain. The electrons go through an electron transport chain, which are a series of proteins. The energy of the proteins are used to pump protons, which are hydrogen ions, to the outer mitochondrial membrane. The electrons are added to other protons and oxygen, which makes the byproduct water. Oxygen that we breathe is moved into the matrix. Protons move through ATP synthase, and they will combine with electrons and oxygen, which makes water. 32 ATP can be made. The electron transport chain is the last step of cellular respiration. NADH and FADH2 pass off their electrons, and oxidative phosphorylation occurs, which is the transfer of FADH2 and NADH+ into FAD and NAD+. Their electrons move through the electron transport chain, and every time they go through one of the proteins, a proton is pumped out because NADH and FADH2 are bringing hydrogen ions. Chemiosmosis is the movement of ions across a semi-permeable membrane down their electrochemical gradient, and a proton gradient provides the energy for the synthesis of ATP. During ATP synthase, protons flow through and generate ATP. Every time a proton goes through, it attaches a phosphate onto ADP to make ATP. Substrate-level phosphorylation is a metabolic reaction that results in the production of ATP by the transfer of a phosphate group from a substrate directly to ADP.
Glycolysis is one of the first steps to lactic acid fermentation, and it yields 2 pyruvate, 2 ATP, and 2 NADH. When glycolysis has little oxygen, it can result in lactic acid fermentation, where NAD+ is required to be oxidized. 2-pyruvate can act as electron acceptors. With this, pyruvate will let NADH know to be oxidized into NAD+, and ultimately reveals 2-lactate as a result.